Dana, thanks for your talk. Dana actually is a really nice collaborator with us. We work together for the short-lived pine tree improvement. And so I'm Chen. I'm the geneticist of the Western Gulf Forest Tree Improvement Program. Today, I will talk about the collaboration, resilience, and the progress. So it's a little bit about the history and the future of the program. For people who don't know that about the program, we are actually located in Texas Forest Service, which is in the university, Texas A&M, at College Station. So first, I will briefly talk about the history of the co-op and the contribution of reforestation in terms of the tree improvement over the landscape in the past 50 years. And then I'll move on to some current breeding research projects with Carol Lubstra, who is here. I know the slide will jump because I didn't fix that. Forest genetics really started by pioneers. About actually more than 50 years ago, Dr. Bruce Zobel established the breeding program in Texas. So he's the first people actually started the landscape level tree improvement program to develop the improved varieties of commercial tree species such as loblolly pine for reforestation. So after Bruce Zobel, Dr. Hans van Buitenen continued the legacy and build up the team. So Tom Byron was the previous director who hired me, and current director is Fred Rayleigh. So due to some unforeseen changes, such as the merging of companies and land and change of the forest ownership, we have a declining membership from about 22 to 13 in the past decades. And I don't know whether the number will decrease in the future. But if you look at this graph, we have an increasing number of contributing and collaborating members. And they are more and more important in the co-op. So USDA Forest Service is an important member like that. Despite of these unforeseen challenges and changes, we are still making good progress in tree improvement. So if you look at this graph, the axis is the years after the establishment of the co-op. For about 50 years, you could see improvement of volume gain from about 5% to 35% till 2019. So this number could increase a little bit now. And if you look at the tree planting area, the acreage in Eastern Texas, which is stabilized around 100,000 acres per year, then try to imagine that if we deploy all these improved seedlings and stalks over such acreage during the rotation time, this improvement could be very significant. So in the future, we want to continue to do that and really keep the goal to advance the collaboration and progress of tree improvement. Also, we want to promote the resilience and the sustainability of both plantation forests and natural forests, as well as the urban forests. So right now, we feel really lucky that we are actually actively collaborating with really nice groups such as Carol's in applying some new technology for this program. So right now, we are integrating the genomic tool developed by the consortium to identify the drought-resistant fast growers in our population. So this study was supported by Arborgen. And interestingly, we found that for the mothers, most of them are as expected based on the registry. But for the fathers, we probably have about 3 or more that are not as expected based on the family registry. And one of them actually is from nowhere in a pollen pool. And the genomic tool at least could help us to quality control the breeding population. And also we could really leverage that to shorten the breeding cycle with good gain in some complex traits such as drought. So why we are doing the drought study, probably it's because it's difficult to do the improvement for this trait as in previous talks, and we really want to tackle that. So that's everything. Thank you so much.
We have time for questions. Any question?
Yes.
How are you evaluating drought tolerance?
So there are different ways. In this study, I think Carol will have more details. So Carol's group is using the carbon isotope to measure the drought response. In my previous studies in poplars, you could actually measure the anatomy traits as well to actually quantify the drought tolerance. Yes.
You guys work with some hardwoods now also? If so, which ones? What species?
Cherry barks.
Cherry bark. Is that the only one?
That's one I'm working on.
Okay.
But Fred is, still maintaining the breeding program. He's doing some selections and tests.
Okay, thank you.
Thanks.
All right, our next speaker is Lisa Tichenko from University of Laval. Lisa will be talking about first step towards unraveling species-wide range genetic diversity in northern red oak for developing a traceability tool. Lisa?
So, hi everyone, I'm Lisa and I'm a PhD student at Laval University. And today I will briefly talk about my PhD project, which main goal is to study the genetic diversity and structure along the natural range of northern red oaks in order to create a traceability system in oaks. So why northern red oaks? Because it is a species that is known for its higher resistance to drought. So therefore it is a potential candidate that could replace certain species that are undergoing some decline in the boreal temperate forests due to disease or climate intolerance. So our first step was to collect leaves or twigs along the natural range of northern red oaks. And on this slide, this shows you our current sampling that we've got so far. To date, our collection consists from more than 500 individuals, comprising 40 population of about 15 individuals each. We have also received some of the sample from other collaborators from the Lobatae sections. And as you can see, we do not have many specimens on the southern range of northern red oaks. So if any of you are interested to help us fill in these gaps, your help will be highly appreciated, because we have we do have some problem because of COVID and collections limitations. So next, our next step will be to extract the leaf DNA and then proceed to DNA sequencing. And after some basic bioinformatic data processing, we will perform some virus population data analysis to identify genetic markers that are related to geographic origin, but also about genetic differentiation at the species levels. And after that, in addition to be one of the first genetic studies in Northern Red Oak in Canada, this will also allow us to create a new reference database. And with those selected genetic markers that we associated with this new reference database, we there will be an excellent tools to for attempting to create traceability systems from matching individuals or wood sample to their geographic origins. So that's it for me, and I hope that the next time that I will be presenting at another conference, I can show you some results.
All right, we have plenty of time for questions. Any question?
So while you're doing this genetic diversity, it seems to me your sampling puts you in a good position to look behind the landscape genomics set.
Sorry, I didn't hear all the questions.
Because of the way you're sampling
Yeah,
to look at genetic diversity,
yeah.
your project is in a good position to do a landscape genomic analysis of Northern Red Oak.
Yeah,
Have you thought about doing such a study?
yeah. I'm interested to do it. So, but because I'm at the beginning of the project, I'm a bit of searching what to look forward. So yeah.
Question? Yeah. You have can you share a protocol for making a collection?
Yeah. I've also brought some kits on how to collect some samples, to show you how it works. And I've also been into the field to collect some samples, so I can also talk and share a protocol. Also, I can make one.
Yeah, I'd like to order a couple.
We have one more question.
Okay.
Can you explain, please, a little bit about what you mean by traceability?
Oh, okay. Traceability is to follow an item or product through the chain of custody, and in forestry, it's more about to identify taxonomically. It could be to be identified taxonomically a species, like an individual, but also to identify their provenance of origins.
Okay, so to do that, you'll need a very extensive sample, right? And you need things from far away.
Yeah.
Can I recommend to you that you talk to a professor named Richard Cronn?
Yeah.
C-R-O-N-N. He's a Forest Service scientist, and he was not able to attend today. But he has developed a system of citizen scientists to collect samples for him. And it was extremely successful. He was able to obtain, for many species, several species, complete range-wide collections. And it was all done by citizen scientists.
Okay.
And so he has a group that he works with already. And he can put you in touch with them. And it would be very easy for them to send you samples from all over the range of red oak. they would be sending single samples, not populations. But if your goal is to represent the full range of red oak, that organization of citizen scientists is a good one. So Richard Cronn, C-R-O-N-N is his name. And I would recommend that you talk to him.
Okay, thank you.
I just want to come in this as a thought, actually. We have been in touch with it.
Okay.
They can actually there's a huge cost. I think it's worth it. But as we say, we are a bit late. We didn't know them.
Oh, yeah.
But actually, this is perhaps it's a non-profit organization, but
Yes.
still we need a lot of money to make business with them. It's citizen scientists, but we train the people.
Yeah.
But yes, this is a huge cost.
Oh, I see. Okay. I didn't know. Thank you.
All right, next, you have a question?
Just have a quick question. Are you, in parts of the range, red oak hybridizes with others, especially in the southern part of the range? So are you concerned, like, is that a problem in the sampling? Like, or like, can you deal with hybrids in your analysis? Because there's a lot of hybridization, like in Indiana where there's scarlet oak and all these other oaks, there's hybrids, a lot of hybrid trees.
Yeah, I know that there is a lot of hybridization, but I don't know if I can answer your question.
Okay. If you need state contacts in the US, like there's specific states, just Natalie knows where to find me. I can get you some contacts.
Okay, thank you.
And they're foresters, they know what to look for. No, they don't require, you say red oak, and yeah, they'll find them. Good luck.
Thank you.
Thank you, Lisa.
Thank you.